Good afternoon, everyone. You are really welcome to this webinar. My name is Adam Kalinowski, and I'm the president of Dystonia Europe. And I'm also an dystonia patient. Dystonia Europe is a pan-European umbrella organization for the national dystonia associations in Europe. But we know we have followers from all over the world. We launched the series of webinars in 2020 to give you the opportunity to learn more about dystonia and the latest news, and also ask questions to specialists. This is the 9th webinar in this series. And so don't hesitate to say hello in the chat and say, where are you from? And you can still invite others to this webinar by sending them a link that you used for joining. So today we have a very important topic to cover. So I'm not wasting your time anymore, and I'm going to hand over now for a few opening remarks to Merete, who will introduce you to the topic and our great panelists. And enjoy our meeting. Merete?
Thank you very much, Adam, and hello, everyone. My name is Merete Avery, and I'm the operation manager of Dystonia Europe, and I'm also a dystonia patient. It is very nice to be here with all of you, and I'm delighted to introduce today's topic, current dystonia research projects. I think that's a topic that's a lot of people with dystonia is very interested in. Sorry, and the webinar title is Exploring Dystonia Pathophysiology Using Rodent Models. Current projects in European Consortia for Dystonia Research. At this webinar, we will have two speakers, and that is Professor Jens Volkmann at the University Hospital in Würzburg, and Professor Wang Ip, who also is a neurologist at the University Hospital in Würzburg. And I want to give you some practical information, and that is if you have any questions, please put it in the chat, because later in the webinar, we will have a question and answer session. And then we will answer as many questions as we can. Then I will give the word to Professor Jens Volkmann.
Yeah, thank you, Merete, and warm welcome and hello from Würzburg in Germany, Bavaria. My name is Jens Volkmann. I'm a neurologist. I was trained in Düsseldorf, New York, and Kiel University. Worked there for about 10 years with Günter Neuschel, a rather well-known movement disorder neurologist, and then moved in 2010 to Würzburg, where I'm now the head of the neurology department, the chairman. And my focus is movement disorders, and in particular the treatment of movement disorders with deep brain stimulation. So what I would like to introduce you to in the next 20 minutes or so is the kind of research agenda that we started in 2010 when I became chairman of the neurology department here in Würzburg, and sat down together to implement a research agenda. We started basically with, identifying a few problems in dystonia research. Dystonia research has been problematic for many, many years because there are several obstacles in disentangling this mysterious movement disorders, as it was called by David Marston, the famous neurologist from London, David Marston. He said dystonia, it was the latest discovery of movement disorders. It was basically his achievement that dystonia was in the 1970s recognized as an organic disease, not a psychological or psychiatric disorder. And it was basically put into the context of other basal ganglia disorders, diseases of the central nervous system. But the main problem in dystonia research is that dystonia is not really a single disease. The term dystonia rather describes a movement disorder which can be caused by a variety of different etiologies. Dystonia can be genetic, as you know. It can be drug-induced, as in so-called tardive dystonia. It may be the consequence of brain injury or brain trauma, or it can be idiopathic, as we call it, which is just another word of we don't know why it happens, which is the case in many of the adult onset dystonias like cervical dystonia, Mesh syndrome, blepharospasm, etc., where we cannot identify a single unique cause of dystonia yet. And all these dystonia diseases, as I would put them together, all these different dystonias, they're still together, they're still rare, which means that splitting them into the different etiologies to investigate unique causes that can contribute to dystonia makes it even more difficult to identify sufficient numbers of patients within a single center. So a single even a large movement disorder center does usually not have enough cases to conduct meaningful clinical studies. And this was the case in particular when we started to investigate the impact of deep brain stimulation on dystonia in the late 1990s. And the first reports came up about a beneficial effect of palatal neurostimulation in dystonia. In Germany, several neurologists and centers of DBS sat together to quickly design a clinical trial, a multi-center clinical trial, because it was very clear that all over Germany, there would be maybe 100 to 150 patients that were severe enough affected by dystonia to be candidates for an initial clinical trial in generalized and severe segmental dystonia. And then one of the main obstacles for understanding the causes of dystonia is that we don't have good disease models, animal models available in which we can study the mechanisms underlying dystonia. Because animals do usually not develop dystonia-like symptoms spontaneously. And even genetic models, as it will be pointed out later by Wang, may have the gene causing the disease, the same gene defect, artificially introduced, as you will find in humans, but the animal does not develop spontaneous dystonia symptoms, unlike humans that have the gene. So all these are a lot of research barriers in dystonia, and it was very clear to us that they could only be addressed in a rather systematic and networking-like approach. And for this very reason, we applied in Germany under the leadership of Christine Klein from Lübeck and myself for a network for funding for a German Dystonia Research and Therapy Consortium, funded by a call of the German Ministry of Research and Education for networks in rare diseases. And in 2015, we were lucky enough to win this call, and the German Dystonia Research and Therapy Consortium, DYSTRACT, was implemented. And this consortium, which was funded for a total of 4 years, contained several sub-projects, some of them very important for subsequent funding. One of these projects, among others, was the German Dystonia Registry, including a gene and biobank. And then we were also able to start funding for a clinical trial, which I will introduce in a second. Subsequently, we used some of these initiatives that were funded by the Dystonia Consortium, such as, for example, the German Dystonia Registry, to apply on a European level within the European eRare initiative for funding for another consortium on a European level, which is specifically dealing with the causes of dystonia. Causes means the so-called pathophysiology, which is the way that within the brain, by network changes or molecular changes, dystonia is provoked. And associated to these research lines are two other major fundings that we were able to obtain. And here we are talking about funding for each initiative in the order of 1.5 to 2 million euro. For the German Collaborative Research Center 295, which is associated with dystonia research, we are even talking about funding in the order of 13 million euro. So based on the previous work and successes that we had in a stepwise approach to the pathophysiology of dystonia and dystonia research, we were able to also obtain funding for another collaborative research center in 2009, together with the Charité in Berlin, where we are specifically analyzing mechanisms of deep brain stimulation and try to optimize deep brain stimulation based on the pathophysiology of different diseases. And one of the model diseases that we are studying within this collaborative research center is dystonia. And then finally, I will also introduce a clinical trial, which then resulted out of these studies, which is starting now in Würzburg and is comparing a new method of programming deep brain stimulation, where we use computational models to identify settings for dystonia patients. And we do this by a kind of an expert approach using artificial intelligence. And we compare the currently standard clinical programming of dystonia versus programming choices that are obtained by this expert system. So let's start with introducing the Dystonia Consortium, which had a 4-year period of funding. And these are the investigators of the Dystonia Consortium here. The principal investigators were Christine Klein and myself in Lübeck and Würzburg. And we were responsible for different sub-projects, but the research within this network was coordinated. And the type of research within this network was spanning from basic research in animal models and cell models of dystonia to clinical application trials, such as the StimTox cervical dystonia trial. Katja Lohmann was an expert leading a consortium on the genetic of dystonia. Christine Klein was responsible for building up the German Dystonia Registry, which now contains more than 2,500 data sets of patients with dystonia, including the biosamples of these patients, which mean we have blood, we have the genetic information, we may have cells, fibroblasts, and these cells and genetic information can be used, for example, in other projects, such as in Project 4 here by Philipp Seipler in Lübeck, where patient-specific cell models of dystonia are being created, which means we can reprogram and reproduce cells from the fibroblast tissue samples that dystonia patients have given, for example, skin samples. And then using specific methods, one can reprogram these cells, for example, to grow nerve cells. And we can study specific biochemical pathways in these nerve cells, comparing nerve cells that were derived from patients with dystonia their skin biopsies and, for example, normal subjects. And this may help to identify molecular causes of dystonia. It may help to identify what is going wrong in the metabolism of cells in dystonia patients. For example, dystonia patients with specific genetic diseases such as DYT1 dystonia or DYT6 dystonia, and compare them to the cell samples of normal subjects that do not have dystonia. Michael Zentner from Würzburg was creating new rodent models with the same genetic mutations in mice that were or that are found and have been identified in patients with specific types of dystonia. And Wang was using these models to analyze system changes, network changes in the brain of these models, dystonia models, rodent models, and he will talk about this line of research later. So just to give you a brief idea of the German dystonia registry, the idea was to systematically collect data on the natural history, the epidemiology, and also the environmental factors of dystonia patients in Germany. And we were able to include more than 2,500 patients. And this database is still present and is growing and is being fed by biosamples, but also by clinical data. And in the meantime, we have also obtained funding from another source for doing genotyping of this entire, or at least a large part of this database, and identifying the number of patients that have known mutations in specific genes that are known to cause dystonia, but also to identify novel genes. And this has been already used in several genetic studies as a sample, either a control group or recently also for genome-wide association studies. And the data is being used not only within Germany, but on an international level. So for the German dystonia registry, we had several centers across Germany, as you can see here on the map. And the 5 main centers were the University of Würzburg, Lübeck, and Hanover, and the Charité in Berlin. And everything was coordinated by our central information office in Marburg. This is a group of Michael Zentner, a neurobiologist from Würzburg, who is a specialist in creating genetic disease models, mouse models. And in his project, he introduced 2 known mutations that are causing dystonia in mice. One was the DYT6 mutation, the TAP1 mutation. The other one was the genal mutation in another form of genetic dystonia called DYT25. And these mice have been created and are now currently being investigated, what is called phenotyping. So one looks basically for abnormalities in their motor behavior, but also on a metabolic level and biomarker level. And then the project of Wang within Distract, and that project was basically based on the very simple idea that not everybody who has a dystonia gene is also developing dystonia. So if you are from a family that has the TOR1A mutation, which is the mutation causing generalized dystonia, DYT1 dystonia, if you happen to have this mutation, your chance of having a manifest disease is only around 30 to 40%. So even though everybody carries within this family, within a generation, individuals may carry the mutation, this is not a guarantee for becoming symptomatic. And hence, there must be other modifiers which basically determine who is becoming sick within a particular generation and who is not becoming sick when carrying the gene. And there are, in principle, two ideas, two hypotheses what these modifiers could be. One hypothesis is that other genes play a certain role in modifying the activity of the TOR1A gene, or it may be an environmental genetic interaction, which means that if you have the gene, you need a kind of a second hit, a second trigger, which is then activating the disease process based on this gene defect. And that could, for example, be an injury, trauma, stress, motor stress, and these are known environmental factors that may or could play a role in dystonia pathophysiology. Wang will in detail talk about this later. Yeah, and an ongoing clinical trial, which was initiated within District, but is still ongoing, has been the StimTox cervical dystonia trial. And why is it called StimTox? Because we are comparing stimulation and toxin-based therapy of cervical dystonia. In this clinical trial, we are comparing the efficacy and also the risk, of course, and the safety of deep brain stimulation and botulinum toxin therapy for cervical dystonia. So you may basically ask yourself, is it useful to have such a trial? Because neither therapy is really a causative therapy of dystonia. They are both symptomatic therapies, but there are certain pros to deep brain stimulation because it's a more durable therapy. It doesn't cause fluctuations. Once you implant the electrodes, you provide continuous symptomatic relief. You don't need repeated injections. And eventually there could be, which needs to be demonstrated by the trial, eventually there could also be a stronger symptomatic effect of deep brain stimulation. So our trial was based on a clinical observation that only approximately 30% of all patients which are being treated by botulinum toxin having cervical dystonia are really entirely satisfied with the therapy. And entirely satisfied in this study, in this Italian study, was defined as having no fluctuations between injections, having no pain from cervical dystonia, being almost symptom-free from the cervical dystonia via the botulinum toxin injections and receiving these injections without adverse effects, for example, swallowing difficulties, hoarse voice, etc., etc. So this apparently, according to the survey, is only the case in 30% of the patients undergoing botulinum toxin therapy. And this for us was reason enough to investigate whether, as an alternative, if the patient wants to undergo deep brain stimulation, in principle, if potentially DBS provides a better control and higher satisfaction in patients with cervical dystonia. And of course, it is very difficult to control bias in such a trial. If we would just operate all the patients that want to be operated and then compare them to the patients that are satisfied with their cervical dystonia control with botulinum toxin, we would get no unbiased answer to this question. So what we designed is a trial in which patients that want to be treated by deep brain stimulation volunteer for a clinical trial in which they are being implanted by deep brain stimulation. And then for a period of 6 months, they receive ongoing injections with either botulinum toxin or placebo, saline, and then in one group. And another group of patients is receiving activated deep brain stimulation. So basically all patients are being implanted with deep brain stimulation, but only half of them is really switched on and is receiving DBS. And this group is receiving additional saline injections every 3 months. and one group is not being switched on with deep brain stimulation and is continuing to receive optimized or adapted botulinum toxin injections, but neither group knows about their group assignment. And this kind of design, which is called a so-called double dummy design, allows us to control the bias, the expectation of patients towards a certain therapy, and to objectively compare the effect of deep brain stimulation and botulinum toxin therapy on quality of life, on symptom control, on pain, etc. The trial is still running in Germany, and we have recruited now approximately 1/3 of the required patients. And finally, we used, as I said, some of the results of the previous funding from the Dystonia Consortium to apply on a European level for the Eurodiscover Consortium, which is a consortium funded by the eRare initiative of the European Community. And it is addressing in preclinical, but also clinical research, the pathophysiology of dystonia, which means the question of how the symptoms of dystonia are caused by brain functioning abnormalities. And this is done in patients and also in biological model systems in a combined approach of studying biomarkers such as cellular biomarkers, but also the effects of deep brain stimulation or recordings from the brain and effect of transcranial magnetic stimulation, modification of circuits. And these projects are all networked and communicating with each other to foster basically with their results, the research, different research lines. A quick look on a consortium that was initiated between Berlin and Würzburg, which is called Retune, and specifically addresses the question of how DBS works, deep brain stimulation works in dystonia and other movement disorders. Again, this is a large-scale approach using different methodologies and technologies in animals and in humans. It's a large scientific consortium. And the vision and the idea of this project, which hopefully will run for a total period of 12 years, is characterizing the brain network abnormalities in different movement disorders, among others dystonia, and then using novel technologies to modify the abnormalities of brain network activity. This can be done, of course, by electrical stimulation, such as deep brain stimulation, but hopefully in the future also by non-invasive stimulation methods, transcranial stimulation, ultrasound stimulation, or in the future, maybe even using more modern approaches of genetic therapies such as pharmacogenetics or optogenetics. And one approach on a network level that was associated with the research on neuromodulation that is being part of this Retune consortium is a study by Martin Reich that already transformed into a clinical application. Martin Reich and his team, who has, by the way, been selected now for the David Marston Prize of Dystonia Europe. Martin has recently published a study in which he reanalyzed the data from the German clinical dystonia trials on deep brain stimulation, and he collected from different centers a total of almost 100 patients that had undergone deep brain stimulation and had neuroimaging information on the location of their electrodes and their MRI. And he used this together with the outcomes of these patients, the degree of improvement, to first create patient-specific models of the electrode location inside the brain. and also models of the electrical field induced by deep brain stimulation. And then these different volumes of stimulation were basically tagged with the improvement that was observed in this patient. Let's say here, for example, 84% reduction of dystonia. And then he aggregated basically the data from all of these patients, and he created a map, so-called probabilistic outcome map, in which he can show areas that produce remarkable dystonia reduction and other areas which produce less pronounced dystonia reduction. And this is color-coded here in this network map. And then one can use this map, which is in reality a three-dimensional map, and one can take specific electrode locations and settings and simulate how much improvement one would likely observe in a patient with such an electrode location and stimulation setting. And in this initial study, we compared basically the model predicted outcome with the really observed outcome after 3 years in dystonia patients. And actually the similarity that we observed between, and the congruence that we observed between the model and the real clinical data was remarkable. Here are 3 examples. In this patient here, 10% improvement was predicted by our model, and the really observed improvement was 12%. Here we see the model predicts 91% improvement. The real observed prediction was 98%. And here we have a prediction of 62 and an improvement of 75%. And all this was enough reason for us to apply for a clinical trial after integrating basically this prediction map into an expert system, which is producing for a given patient with a given electrode setting hundreds of possible or even thousands of possible combinations of stimulation settings within the computer, and then selecting the best options. This expert system is being tested now in a clinical trial where we compare this expert system based on artificial intelligence with the standard approach of clinical programming of dystonia patients. And we hope that we can show that our expert system can provide equally good or even better outcomes at the same, but with a markedly reduced programming time, such that in the future patients with dystonia can hope for a much more reliable outcome and programming based on just visualization of the location of the electrodes. So this was a tour de force through different projects that are being currently conducted in our center in Würzburg and the associated consortia. And I'm happy to take your questions after Wang has introduced more specifically the animal research line.
Yeah, thank you, Jens, and thank you, Marita and Adam, for the invitation to have this talk today. And good afternoon, everyone. I'm Wang Ip. I'm also a neurologist working at the Department of Neurology in Würzburg at Volkmann's department. And I have been working on rodent models of dystonia, as already mentioned, for the last, let's say 8-9 years. So after Jens Volkmann came to Würzburg and took over as a chairman of the Department of Neurology, we started to build this preclinical line of dystonia research because of the problems that he's already mentioned in the field of dystonia research using animal models. And the first step was really to generate an animal model or models for dystonia that show both the symptoms of dystonia and have some genetic background that we can work with when it comes to looking for the causes of dystonia. And let me share my screen now to show you what projects we have. I hope you can see my screen. And now the topic, the title is Exploring Dystonia Pathophysiology using rodent models. And the first part, I would like to give you a short introduction about the evolving definition of dystonia, because this impacts the next point, that is the use of animal models of dystonia, and the evolving definition, the difficult definition of dystonia makes the work with animal models also very difficult. And as a last point, I would like to show you some data about this research line of the second hit hypothesis in dystonia pathophysiology. So the term dystonia was first introduced by Hermann Oppenheim, a German practitioner, who wrote actually a paper describing patients with dystonic symptoms, and he called this disease dystonia musculorum deformans. And this paper became so famous that it was translated then over 100 years ago, so in 2013, and was published in Movement Disorders, and entitled About a Peculiar Cramping Sickness in Children and Adolescents. Now later on, there was a lot of other authors describing the other dystonic patients and trying to characterize and classify dystonia, so the disease and the symptom of dystonia. And what I want to show you here is that there is a developing complexity over time. And the latest definition is shown here on the right-hand side, published by Alberto Albanese and colleagues in 2013, also in Movement Disorders. And as you can see, there are really several lines of definition really pointing towards this complex disease. And because of this complexity of a disease, it's, I think, clear that when you want to work with animal models, it becomes even more complex, because you have to choose what you want to analyze. And basically, one can look for animal models that show the dystonic symptom, the so-called phenotype, or one can use animal models that have a gene defect, an underlying gene defect, as we find, for example, in genetic forms of dystonia, so the so-called genotype. So one has to make a choice. And when it comes to working with animal models for dystonia or other diseases, a lot of people like to work with mice. However, the problem with mice is that the size of the brain is quite small. And in several research lines, the brain of the size really matters. We would like to have a bigger brain. For example, I have a research line where I perform deep brain simulation in rodent models, and there the size of a rat brain is more of advantage compared to that size of a mouse brain. There are even people working with monkeys, and there is a dystonic hamster I want to show you later on. So, and before going to the animal models, I need to introduce another a term that is called validity. That's important when it comes to working with animal models. So this validity term is subdivided into several subterms. One is called phase validity. This basically reflects the symptom that the animal model has, and a good phase validity means that the animal model shows the same symptom as you find in the human disease. In our case, we would like to have an animal model that shows a dystonic phenotype, so dystonic symptoms, similar to what we find in human patients. So the second term is the construct validity. So the most easiest way to describe this is actually that to find an animal model that shows the same genetic defect as we find in human. Yeah, and a third term is the predictive validity, and this refers to the same reaction or medication that you find in the animal model and in the human patient. So I would like now to show you some examples of animal models. First of all, I give you an example of a pharmacological animal model. Then I will show you some videos of a spontaneous mutation, and then give you a short introduction about a DYT1 model, so a model for a monogenic form of dystonia. And the focus will be on phase and construct validity. Yeah. The first model I want to introduce to you is the kainate dystonic rat. So it's a pharmacological model. Kainate is it's a glutamate receptor activator. And what you can see here is that after injection of the kainate into the brain of the animal starts to develop over time an increase in dystonia. As you can see here, there is a first of all an extension of the tail. After 16 hours, it becomes worse. The hind limbs are involuntarily stretched, and the animal cannot move properly. And the dystonic symptoms become worse and worse. And this has been published by Alvarez, Fischer, and colleagues in 2012. And what they found out was that in this animal model, because of the kainate injection, there is an increased activity of the neurons in several regions of the brain, such as in the cerebellum, so the back part of the brain, but also in some other regions. While in the control animals, we do not see such activation. So this shows us that the phase validity of the animal model is very good, because if you find symptoms that are dystonic or dystonia-like, yeah, it's so good that this model has been transferred to other animal species, such as to mice and also to cats. Yeah, here you see a cat with actually cervical dystonia after the injection of the kainate. However, the problem is that there is no genetical relation to human dystonia, so the construct validity is not good here, okay? And this makes things difficult and problematic if you want to analyze the underlying cause of this problem on a molecular level, for example. Yeah. So this is the next model, the dystonic hamster. Yeah. And as you can see here, DT is the abbreviation for dystonic, and SZ here is actually the abbreviation for seizure, because when this model came up just by chance, because it was a spontaneous mutation in Syrian golden hamsters, people thought that this animal model had a problem of epilepsy, so of seizures. However, later on, more intensive research showed that this model has some dystonia, but no seizures. And one of the pathophysiologic reasons why this animal model develops this kind of dystonia that becomes severe over time is because the animal has a maturation defect of certain neuron population in the brain, in the basal ganglia, the so-called pathophysiologic neurons here. And as you can see here, at 30 days of age, we have tiny little cells here, and they are very small. And later on, after 19 days, you can see that these cells get some ramification, very pronounced. So there is a maturation defect, and this reflects very well also the time course of disease. These animals have dystonia at the early stage, but for example, here after 19 days, dystonia is not present anymore. So here we have an underlying problem. But the problem is that we have a spontaneous mutation, and the mutation itself is unknown. So again, here we find a very nice model with a high phase validity. It's a good model to analyze, for example, dysregulation of the basic ganglia and analyze this model as a network disorder. However, the problem is that the construct validity is low because we do not know the underlying genetic defect. Now here you have two video recordings of two mice, and both of them are DYT1 models. So on the next slide, I will give a few words about the DYT1. What I want to show you here is that both models do not show a clear dystonic symptomatic. As you can see here, the mice run around here and they look pretty normal. Yeah, and although they have an underlying gene defect, and it is the GAG deletion, as we find in patients with DYT1 dystonia, also called DYT1A dystonia. And the underlying gene defect is in the TOR1A gene that encodes for the gene torsion A. Yeah, it's the most common isolated form of dystonia, so a monogenic dystonia with a disease onset in childhood. And we know that torsion A is interacting with a lot of cellular processes, such as a physical function, axonal transport, and on the other side, you have the central motor network. And what's not known is how the alteration of the torsion A encoding gene leads to an alteration of the central motor network and then to dystonia. So we have now this animal model that do not show any dystonic phenotype, dystonic symptoms. So the phase validity is bad. Yeah. However, we know that this model has a known gene defect, and the question was now for us: is there a possibility to somehow change the phase validity to a positive one? So to somehow induce some dystonic symptoms in this animal model, because only if we have really dystonic symptoms, we can transfer this actually the research we are doing on the models regarding therapy to human. And here the idea of a second hypothesis came up, and as Volkmann already introduced this idea, let me give you also a few introductory words about this. So DYT1 is known to have a reduced penetrance of 30 to 40% only, and this means that only 30 to 40% of the patients carrying the G mutation really develop the symptoms of dystonia. And here we have a patient. I cropped this video because of respect of the patient. I didn't want to show the face. I cropped the video, and you can see here, nonetheless, the patient has a severe generalized dystonia. And this is a patient who has a DYT1 mutation, while on this side, you can see his father, who carries the same DYT1 mutation, only has a mild dystonic tremor. Okay, so it's really mild, and you don't see much. So how it comes that there is such a difference in these two patients, although they have the same gene defect? And the question is whether there is a gene-environment interaction could be a trigger of dystonia. And specifically, we wondered whether trauma can be used as an environmental factor to trigger dystonia. Yeah. So the idea of or the connection between dystonia and trauma is not new. It has already been described, for example, here in 1886 by a very famous neurologist, Sir Richard Gowers, who described a naval officer who sprained his thumb and then developed writer's cramp later on. Yeah. Later on, David Marston published a very nice paper describing 414 patients with a cervical dystonia, and in 9% of these patients, he found a preceding trauma to the neck. And he writes here that he cannot rule out the possibility that a trauma might be a specific trigger. And this nerve injury led to transient paralysis of the leg, but then the animals recovered. And this is a video showing you a DYT1 mouse model after the nerve injury and after a recovery phase of weeks. You can see that the right hind limb here shows some involuntary retraction, some cramping, something that reflects, at least for a rodent, a dystonic phenotype. Here's a wild type animal. So wild type meaning that this model does not have any gene mutation as we find in the human, as a control animal, where you see that the cramping is not present or less severe. We perform some scoring of different cohorts of animals and analyze them. And indeed, we saw it here in the gray line that the DYT1 rodents have a higher dystonia-like phenotype compared to the wild type normal control animals here in black. Yeah. And so using this peripheral nerve injury as an environmental factor as a second hit, we could indeed induce a good face validity in these animals and that have a good construct validity per se. So here you can see now the further analysis we did. One thing we did was to analyze now changes, for example, here in the dopamine metabolism. What you see is here analysis of the dopamine transporter protein level and the binding activity, so the functional level in the brain and the basic ganglia of the different mouse cohorts we had. These are the DYT1 animals after a nerve injury compared to the animals before a nerve injury. What you can see here is that there is a dysregulation of the striatal dopamine. There is a reduction, for example, of the dopamine level in normal wild type mice after nerve injury as a, let's say, normal reaction to the injury, while in the DYT1 rodent, there's an increase. Yeah. And here, for example, the dopamine level is also changing. Yeah. In the wild type animals, there is a reduction after nerve injury, while in the DYT1 animals, it stays in the same level. Yeah. So we wanted to validate this finding in a second DYT1 model. For this, we use a DYT1 rat model. And as you can see here, after a nerve injury, there is an even more severe dystonic or dystonia-like movement here in the right hind leg. And here in this model, you can see, in this rat here, you can see that there is even an overflow of the dystonic activity to the left side, although the injury was only on the right hind limb. We again scored these kind of alteration and of movements, and you can see here the DYT1 rat model here has a higher dystonia score until the experiment was terminated at 12 weeks after a nerve injury, while here in green, the animal model, the wild type animals, the ones that did not have the gene defect, recovered completely to a zero degree of dystonia. Yeah. So using this model, we wondered whether deep brain stimulation can be used as a treatment, because we know that deep brain stimulation is a very successful and good treatment for patients with cervical dystonia and generalized dystonia. And so that's why we wondered whether from the pathophysiologic side of view, whether deep brain stimulation within the internal pallidum could also improve the symptoms in these rats. Because if this is the case, then we can continue to analyze, for example, the molecular mechanisms of deep brain stimulation. Yeah, and this is how it looks like when we perform the surgeries. This is a stereotactic frame, and in the middle you can see a rat that is receiving an electrode into the internal pallidum. And this is the setup that we have for deep brain stimulation. You can see the cable. It's a cable-bound setup. We also have a wireless one, and this cable transfers the stimulus, the stimulation to the brain. And this is now after sacrificing the animal. You can see here where the arrow points towards the internal polygone. Yeah, and after 3 weeks of simulation, it looks like this. And this is an analysis here showing that indeed after 3 weeks of polygone stimulation, we have a reduced dystonia-like movement score shown in green in the DYT1 red model compared to the non-stimulated ones here shown in black. So this is just some a bit of an insight of what we're doing. There are a lot more projects ongoing. So what's important to what I wanted to show you is really that the choice of the animal model is very important in dystonia research. And what I hopefully could show you is that dystonic DYT1 rodents show a disturbance of the basic ganglia dopamine metabolism and also respond to deep brain stimulation, and that environmental factors, in this case trauma, can elicit dystonia in genetically predisposed DYT1 rodents, supporting our idea of the second hit hypothesis. Now, this is the group that is working on all these rodent models, and as Volkmann already mentioned, we have received a lot of funding the last years, and that's why our group is really growing over time, and so that we will hopefully see many more projects and also a lot more regarding the basis of dystonia pathophysiology within the next years. So I thank you also for your attention, and I'm happy to take the questions.
Okay, thank you very much, professors, for your very good presentation. I know I can understand that for patients, not everything it's easy to understand, but in my opinion, that was one of the best research presentations I saw so far. It's well done, and for me, it's easy enough to understand what's the research about. So as I can see, lots of questions coming. So please free to take these questions and try to answer if you can.
Maybe I can start with this one question from Richard Werther. Emerita also wrote something. Is dystonia considered a degenerative condition? It's hard to get a straight answer on this, and because from when I worked on the DYT1 model, this was something that I also wondered whether we find some neurodegeneration. And what I didn't show you that is that I also worked on another mouse model. It's a DYT12 mouse model, so for rapid onset dystonia Parkinsonism, another genetic form of dystonia. And so it really depends on what kind of dystonia we're working on. For example, DYT1 is not considered to be neurodegenerative. It's a network disorder where we have some dysfunction in the brain. And what is shown is, for example, that in patients, there are some older paper showing some inclusion bodies within certain regions of the brain, or some changes in the dopaminergic neurons, like enlargement of dopaminergic neurons in a certain part of the midbrain, the substantia nigra, and but no clear evidence for neurodegeneration. However, there is the group of William Dauer, who has generated some other DYT1 animal models by changing the protein levels of the DYT1, and what he could show several years ago is that in one certain DYT1 model, there was a degeneration of different regions of the brain, also, for example, of the cerebellum, leading then to dystonic symptoms. Yeah, but this is something that we do not find in humans. However, for example, if you look for this other dystonia form, the DYT12 dystonia, there are several autopsy paper showing a clear degeneration in the basal ganglia region, also combined with some inflammation, some gliosis. So some of the other cells, or so-called glia cells, are involved, and seems to be some reaction, secondary maybe to the degeneration. So it depends really on the kind of dystonia you're looking at.
But I think, I mean, from a clinical perspective, to add on what Shivang has said, for the vast majority of dystonia patients, the hypothesis is that the nerve cells in their brain are dysfunctional, which means they are alive. They do not degenerate, but they have problems in either the way that they transmit information, that they work electrically or chemically, or in the connection between different nerve cells. So the brain basically is a supercomputer, and the supercomputer consists of, individual nerve cells. And in order to function well, these nerve cells need to work on a single cell level. They have to fulfill their task of transmitting information, but they also need to be wired together in a correct way. And this is what we call a network disorder. And the current thinking is that the dystonia symptoms are largely arising from false connectivity and interaction and information flow between different nerve cells and regions of the brain. So I take another one. I start from the top. I have the question from Timothy Krüger. In your opinion, what are the most successful treatments with regard to dystonia, and what are some of the success stories? Well, of course, I mean, the most successful treatment that is commonplace nowadays is botulinum toxin, but botulinum toxin is not is a purely symptomatic treatment, which is not really addressing the causes of dystonia. It's basically treating dystonia by denervation. So it's treating the transmission between nerve and muscle, the information flow, but this is not where the dystonia problem is actually located. So it's a successful treatment because it can reduce the spasms, the dystonia, the abnormal movements, but it doesn't address the cause of dystonia. And in terms of addressing the cause of dystonia, deep brain stimulation is actually the most successful story because it is directly interfering with the abnormal network activity, the information flow, the dysfunctional information flow between different brain regions. And the success stories of deep brain stimulation, and we start to implement, to understand better and better how deep brain stimulation works, where it needs to be applied, how critical it is, where to place the electrodes to get the best possible effect, and also to understand how we can predict, for example, with these artificial intelligence algorithms, outcomes of DBS in dystonia patients.
Maybe to add to this, I think another very successful treatment is levodopa for the levodopa sensitive dystonia. So because there are certain forms of dystonia where we have some deficiency in enzymes or enzyme activity that are somehow dopamine related. There are several forms of dystonia that, so for example, DYT5 dystonia that respond on levodopa, and this is something that really can improve the symptoms very much. And I think this is also something that is as we mentioned.
There is another question from Stuart from Scotland. He is asking what research is there currently being carried out into the use of transcranial magnetic stimulation to treat cervical dystonia. it's more a research tool, transcranial magnetic stimulation, than really a therapeutic tool, because the currents that are induced in the brain are too weak to penetrate into the depths of the brain. And it is currently believed that most of the problems in cervical dystonia are resulting from dysfunction of either the cerebellum or the basal ganglia, or the interaction of both. So to basically stimulate in the depths of the brain, the cerebellum or also the basal ganglia is very difficult with standard transcranial magnetic stimulation techniques. And it rather requires invasive stimulation, such as placing an electrode in these areas and then studying the effects of focal stimulation with sufficient strength. So I have my doubts that transcranial magnetic stimulation will ever be will ever have a therapeutic impact in clinical practice. There is another DBS-related question, maybe how sensitive, from Richard Werther, how sensitive are DBS outcomes to the location of the wires? Location is the one critical point of deep brain stimulation. And so we are talking about stimulating only a few cubic millimeters within the brain. And an electrode that is placed only 2 millimeters or 3 millimeters away from the optimal target will have an inferior result. And we know this because we have in patients with Parkinson's disease, where the critical area is even smaller, we have conducted revision surgeries to replace electrodes that were 2 to 3 millimeters off target, and have obtained much better results and outcomes after replacing the wire. So really the location of deep brain stimulation is critical, and the location of the electrical stimulation field depends A, on the electrode location, and B, on the settings. Because with the parameter settings, you can influence also the area that is being stimulated. But usually we are talking about a few cubic millimeters and distances of maximum error tolerance of 1 to 2 millimeters. Everything outside of 1 to 2 millimeters may be off target.
Okay, the next question is then from Facebook. Why Botox injection lifetime is very small? So I think it's one of the reasons, of course, the way botulinum toxin works. I mean, it affects the neuronal cells that are connected to the muscle. And of course, these cells are still viable, so they are still alive. And what they do is, although the link to the muscle is then broken by botulinum toxin, the cells can still sprout some new connections. Yeah, and this is something that takes around about this time of 3 months or so, and that's why it's always the problem that the duration of the botulinum toxin effect is around this range. Yeah, maybe the next question: why new treatment is not coming? We're facing so much struggle. Yeah, this is really, a problem, and I think what we could show you is that we are trying from our side, at least, to tackle the dystonia disease from both ends. So on one hand, we are working on the things that are already known, like deep brain stimulation, botulinum toxin therapy, to try to improve the therapy there and to get more knowledge about this. And on the other hand, we're trying to go to the basic research side, trying to find, for example, new molecular targets. For example, this project that Jens has has talked, or this problem, we are trying the molecular changes of dystonia using different models for this culture models, and then also biosamples from patients such as blood and blood cells, and trying to figure out what common changes we find and what different changes we have, so we might find some relevant new targets to work on. But these are things that take really a lot of time and a lot of work. That's why it's so difficult, also because dystonia is such a heterogeneous and very rare disease.
I must say, I mean, new perspectives are really resulting from these new research lines. For example, the idea that a gene defect is not, necessarily sufficient to cause dystonia, which is the case in DYT1, where you can have the gene, but almost 60% of the patients having the gene do not develop dystonia. So, I mean, one can see it from the perspective of, oh, these are the lucky ones and they don't get the disease, but, genetically they have the disease. So the real question is, why does the brain of these, individuals not develop dystonia? And this is maybe not the case because the gene is not, the disease process is not strong enough in them, but it could be compensatory mechanisms. So maybe these individuals are fit enough, their brains are fit enough to fight the dystonia so that it's not manifesting itself. So they have some, either on a cellular level, metabolic level, biochemical level, they have some unique features that do not allow the dystonia to take over in their brain, simply spoken. So new treatments could also come from learning from these mechanisms, how the disease is being suppressed in those patients that have the gene but do not basically develop dystonia. And it does not necessarily need to be a treatment on a genetic level. This could be, other factors that are being supported, enhanced to basically fight the dystonia mechanisms. And it could also be a preventive treatment. If we understand, for example, the mechanism of these triggers, if the second hit hypothesis turns out to be true, that you need a gene and then an environmental cause, which is then activating basically the disease process, then of course we could potentially have preventive treatment in individuals that are being identified very, very early on as gene carriers, and we could basically either treat them pharmacologically or by some other measures and help them not developing dystonia, although they have the gene. So all this is maybe different from the idea of having, just one medication that you take or, just one pill that cures dystonia. But these are different options to at least help manage dystonia.
Okay, there's another one. What are the plans for the research project after the 3 years are ended? So luckily we are really at the beginning of nearly all of the projects that are running. And for example, this European Consortium that Jens has talked about just started a year ago. And if we're lucky and we can then extend this up to 12 years, the Eurodiscover project is really something that's only lasts for 3 years. But we're hoping to achieve so much success here in the outcome of the projects that we can then apply again as a consortium for next funding, not the funding period, but for another funding. For example, there is another European funding called what is called just Dystonia, but not Horizon 2020. What's the it was formerly called Horizon 2020. I think it's called Horizon Europe now, and this one starts to have a call on rare diseases in 3 years. So this means that after completing the Dystonia, the Eurodiscover project, we can try to apply the consortium for this proposal, for example, for this call. Yeah.
And it's very important that we have the support of patients for these kind of projects. I mean, in particular in clinical research, it is important to have access to patient data, to biodata, which means that, the more patients basically engage themselves in registries and clinical trials, the more data will be available for conducting and testing research hypotheses. So Dystonia Europe is actually part of the EuroDiscover consortium, and patient advocacy groups play very well to the affected patients, but also to help recruiting patients for this kind of research initiatives.
So there's a question of whether Botox, because it contains protein, is possible to induce kidney stones. I'm not aware of this as a side effect of Botox. I mean, it's a foreign protein, that's right. It's very well tolerated. I mean, by injecting foreign proteins, it's always that the question whether there is a sort of an immunological reaction, to it. But this is something that has been investigated very thoroughly, and it's rather low. But kidney stones is something that I'm not aware of, that this is a main problem.
Yeah, then there is the question of, could vibrotactile stimulation be used for, that is used for Parkinson's disease, possibly work for dystonia patients as well? Yes, I think so. I mean, you're all aware of dystonic tricks. Touch to specific areas can be used to overcome dystonic movements. So that potentially a kind of a jest or tick maneuver could be replaced by tactile somatosensory stimulation. So that's entirely possible. But it would be very individual for each and every patient, because not every patient has the same trick, as you all know.
Okay, there's a question from Jill Olsen-Moser. When you talk about trauma as a trigger for dystonia, would this trauma always be in the same area where dystonia develops? Speaking primarily of task-specific focal dystonia. This is something, it's a very interesting question. And for example, in the case of the animal model, I think I could show you that there were some hints for a spreading of the symptom, also to other parts of the body. And I believe that this is something that is also happening in patients. So this, this change of plasticity and loss of inhibition in the brain regions that does not usually only cover one part of the body, but it can spread also to other regions.
Yeah, I mean, when we talk about trauma, we really talk about physical trauma, not psychological trauma. This is another question by Jill Olsen-Moser. So there is currently no evidence that stress or stressful experiences or traumatic experiences could trigger the onset of dystonia. What we are referring to here is really physical stress. Trauma, overuse, excessive use, for example, painful use with some, tendovaginitis, inflammatory changes in the peripheral tissue that cause pain, for example, in writer's cramp. These kind of stressors, physical stressors, have been related to dystonia, not so much psychological trauma. And yeah, Adam is asking if, for example, stress activates the dystonia gene. Is there any chance that the symptoms will go away in the future? Unfortunately, I think this is a kind of a hit and run mechanism. So you're hit once, the stressor goes away, but you remain with the symptom. It triggers a change in the brain that is largely irreversible. And this is why we seldom see spontaneous recovery in dystonia patients. In general, dystonia may fluctuate, of course, but in general, clinicians would say that a dystonia that is present for more than 12 months is very unlikely to go away, whatever the cause of dystonia may be.
So next one is Vas Angel. So two questions, but I think your question is, I was told, so is it possible, are there tests that one can do to determine dystonia? So I think dystonia is, generally speaking, I mean, it's a clinical symptom and clinical diagnosis. Of course, there are possibilities to do some gene panels, some gene testing to see if there is an underlying genetic cause. But of course, it's only the minority of genes that we know at the moment. There are for sure, there will be much more genes over time. Once we know more about the different types of dystonia, and not sure if there's anything else one can do. Yeah. Anything from your side, Jens?
Well, functional dystonia exists. It's basically a false body representation, as one would call it. So it's basically a movement disorder that is caused by a false concept of your body representation. But in contrast to an organic dystonia, exercise and or psychotherapy may help to improve that kind of dystonia. So in the end, this is really the best way to define functional dystonia from the perspective of treatment. If it can be treated by particular exercise programs and psychotherapy, then it is likely to be a functional dystonia. Initially, to distinguish an organic dystonia from a functional dystonia is quite difficult, and it requires, I would say, expert knowledge and skills. Is there any research in the concept of abnormal connectivity that could help to improve the treatment? Is a question of Ophelia Cruz. Yes, I mean, that's actually very tangible. I mean, with perspective to DBS, connectivity analysis are currently, right as we speak, basically, are being used to analyze basically the network that is influenced by specific electrode locations. And then again, this information can be used in individual patients, for example, also within the surgery, to define whether a particular electrode location is really addressing this network. This can be done, for example, by functional magnetic imaging. A study in Toronto has shown the feasibility that within the scanner, one can stimulate through the electrodes electrically and then look at the brain responses in the fMRI. And if you see a certain pattern of interconnected regions lighting up, then this means or is a biomarker for a good outcome of DBS and dystonia. And these are, avenues that are being explored currently, that one can better predict basically the therapeutic impact of DBS in a given patient without waiting for, sometimes weeks and months to see the expected result.
Jens, there's a question about dystonia expert in Portugal. I'm sure that you know someone, right?
Yeah, we have been working a lot with the center in São João in Porto, where I've been visiting basically for the last 17 years to introduce deep brain stimulation for dystonia in Portugal, and the neurologists there are quite knowledgeable in dystonia. Also, the treatment with botulinum toxin or with deep brain stimulation.
So I just want to mention that on the Dystonia Europe website, we have created this year a page with dystonia specialists worldwide. But I just checked out this list and we have no Portugal there, unfortunately. So if you can send to Dystonia Europe details of this hospital where
Sure.
people can get a treatment, we can add this to the list. And for our listeners, if you go to Dystonia Europe website, you can find this organization worldwide on our page. And we will add the Portugal as soon as we will get it.
There's one timely question. How long do we have to wait for Botox injections when you have COVID-19 Delta variant? So that's a timely question. We are all concerned about COVID. I think, with respect to the botulinum toxin injection, it's basically a question of how long you are infectious. If you're not infectious anymore, you can get your botulinum toxin injection. There is no reason to wait. There is no interference between COVID-19 and botulinum toxin pharmacology. But of course, you should not infect others, which means that you have to wait usually the 2 weeks after the infection and a negative test. So that the quarantine period basically, and to have a negative PCR test in order to be admitted to an hospital again, or a private practitioner, private practice that can then conduct the botulinum toxin treatment.
All right, and I think there were many of interesting questions, and you answered for most of that. And also some questions were covered in your presentations. So, okay, thank you very much for that. And now, Merete, do you want to say a few words of summary?
Yes, I will try. It was a very highly scientific presentation, but very interesting, and yeah, I'm impressed by the work you do, and I'm also very thankful to Professor Jens Volkmann and to Professor Xing Wang for the work they do, and it also puts a light on how hard it is to find all the details and find out everything about this condition. And I know that research takes time, and I'm saying this to all the viewers because we do want fast answers, but it does take time. And also, I think it was a lot of great questions, and some of them was very, the answers was really good, because a lot of people think about, I have dystonia, and I have children, and but as it was said, mostly it is not passed on, which is really good to know. I can also say that from my own family, my father has it. We have all my uncles had it, and none of my cousins from all the 8 uncles and aunts does not have it. So I want all those who worry about that to think about that only 60% of patients that have dystonia that is genetical does not develop dystonia.
In my case, I can say as well, nobody from my family for generations and now had dystonia. Just myself. So there is a little tiny chance. So this is a good news.
And then, of course, a lot of people just has a dystonia, which is not genetic. And I understand a lot that a lot of people struggle with the treatment, but seek a second opinion. And I'm just kind of, I think that all the information we got today was a little over, is a lot, but we can see it again. And it's great information. And we're so thankful for the research that's being done. And I hope that everybody who viewed this in the future and viewed it today realizes how difficult it is to really solve this.
And I'm always saying that in case of dystonia disorder, there are more questions than answers.
Yeah.
Okay.
Yeah, very good.
So let me wrap this meeting. So I think it was extremely informative webinar, and I hope that maybe we can repeat that in the future with an update on the research, maybe. I want to thank to all of you for joining and interacting. I want to thank to our specialists for your time, support, and work you do every day. And thank you to Merete and the whole Dystonia Europe team for their work, and our partners who have been supporting us to facilitate this webinars project. And I want to inform you that a recorded version of this webinar will be available on the Dystonia Europe YouTube channel and Facebook. So if you want to watch it again or share it with others, please follow our social media channels that will be available soon. And we also have a lot of material on this topic and different dystonia-related topics on our YouTube channel. So it's really worth to take a look. So thank you for taking the time to come to this webinar. And we look forward to having you at our next webinar, which will be held in September after the summertime. So in the meantime, I wish everybody a nice summer and good evening. Bye.